Hey everyone, I hope you are all able to hear me and listening in to our next SCOG single cell Omics Germany network virtual lecture event. I think that's about to have Prisca Liberali talking to us today. Before we get started, I think there's still people joining. You know how things, thankful that you come to this meeting. Very happy about that. We do this every 2 weeks. Try to check out the next ones. There's also a very nice workshop from Young coming up on recent advances in single cell epigenomics. I think a lot of exciting things happening on that side. Although, of course, we just had a chat before with Prisca and others. We kind of said that we don't do this physically. We would love to see you all and I don't know, have a coffee, because this is the morning or in the afternoon, starting to get drunk as we usually do at those meetings. Well, but yeah. Having said that, I'm very her nature intestinal organoid variation. This is like such a cool data set. I saw her present this was like 1.5 years ago, I think at Keystone. And this was one of the major talks at the meeting, I felt. And really excited to hear about new things she's cooking up. Super shortly about her background. Prisca is chemist by training, doing then PhD in biology, following up with a postdoc in molecular systems biology at the ETH in Zurich. Essentially with a junior group since 2015 at the Friedrich Miescher in Basel, together with a professorship there. Today, she'll be talking about lineage tracing of stem cell population dynamics, and I think it's really fascinating how she combines really nice biotechnology with deep biology questions, and then also very nice and interesting analysis, also network systems biology analysis. Really looking forward to talk. Thanks for coming, Prisca.
Okay, thank Fabian for the introduction. Thank you all for joining this talk, this virtual talk this morning. So today I will discuss how we'll use image-based technology to try to understand how tissue develop from single cells, or how they regenerate from adult stem cell. And I think one of the main challenge we always had is that tissue are not constructed machines. They don't have a blueprint. So what they are built bottom up, with individual cells changing shape and function, acquiring a really new function and genetic program, while always maintaining a continuous communication with the other cells. And so this is inherently a very complex process that is genetically wired to keep it precise and reproducible. However, we need to have a certain robust and contextual, maintain this space-time coordination with the rest of the, well, what we call the design principle of tissue organization. It's really how single cells coordinate across the scales, both spatially and temporally. And we use the mouse intestine to, and also the human in certain cases, but this will be mainly on the mouse intestine. And the mouse intestine is very spatially organized with the crypt bottom, where stem cells and panet cells, these are differentiated cells that secrete wind and maintain a stem cell niche, the transit amplifying cells, and then the enterocyte, that are the mainly absorptive cell in the intestine. What is really most amazing about this is that these tissues can be recapitulated in vitro. So already in 2009, Hans Klever showed that a single LGR5 positive cell is able to form an organoid in something like 5 or 6 days. And this is quite an incredible process, where a single individual cell is able to divide. In these first 3 days, when the cells are in Win3A, there's what we call a symmetry breaking and differentiation. So you have the formation of the first panet cells that are the first event that then create the stem cell niche. So at a certain point, what is this is that how does an individual cell sense the extrinsic cue from the environment to take individual decisions, but coordinated decisions, because you don't have multiple symmetry breaking events, you just have few that then create the stem cell niche. This is a problem about biological scales, in the sense you have very fast millisecond events on a plasma membrane of this individual cells that have consequences days later on the tissue level scale. And this was what we were interested in. And so what is our main problem is that we are interested in decision making, that is this area here. So we use single cell RNA-seq to find the different cell type, we can start inferring trajectory from also this data. But in reality, we don't know what is the exact individual cell trajectory inside these population in a multivariate feature space, especially because when you start looking at these transit amplifying and this progenitor population, these are extremely variable, but this variability is very slight. And so the main problem is that we miss spatial context because these decisions are very contextual. They depend on the microenvironment, about mechanical properties of the environment and of the tissue, about the cell-cell communication. Because what we know is that it's not only the RNA as a messenger that is making the cell take the decision, but of the protein and the phenotypic cellular state. So what we mean is that in this population of cells, so these cells that make decision, these are different cellular states. And these cellular states don't have a random intrinsic variability, but these are metastable cellular states in their microenvironment. So we would have subpopulation of cells that are, for example, in a certain cell cycle state or in a metabolic state in that population that have a higher probability to differentiate and maybe take one certain direction in this trajectory and are very early on. And it has been shown also by Alan Klein recently that even when you put barcodes and you do the full lineage tracing, you have a hard time really predicting very early on what will be the decision of the individual cells. And there are things inside a cell that cannot be measured by RNA-seq. And the other one is that stemness is really a property of a population of cells. It's not an individual cell that self-renew and differentiate, it's a single cell in a population. And this, I think, is very important because we need to be able to measure the individual cells, individual organoid in many different organoids. And so for this, we use two different main approaches. One, we use 100,000 of individual organoid with a very high dimensional measurement. So we can use multiplexed, and then I will come to that imaging. And on the other side, we use a much limited number of organoids, but we look at dynamics for days. So to look at this, the first part, what we do is start from a mouse. We take intestinal stem cells, encrypt from there, we make organoid, and then we sort individual cells. We take LGR5 positive, so stem cells, and also LGR5 negative cells, because the intestine is a very plastic system where also LGR5 negative, so non-stem cells, are able to form organoids. We put them in multi-well plates, and then we fix them at different time points. What we use is this multiplex protocol that was developed in Lucas Feldman's lab. This is a really important step in our acquisition stage, because what we do is that for the same individual organoid, we don't only measure 2 or 3 fluorophore, but we can go up to 12, 40. Okay, we don't get to 40, but this is in theory possible. So what's happening is that you have your sample, you do a normal immunofluorescence, and then from there, you have your image and acquisition, and then you can elude the antibody, like really stripping it from the images and restarting from scratch. Here you see, for example, one example where you have the same organoid 12 times, so with 12 multiplexed rounds. From this, we then use image analysis and different machine learning and to look at the different cells, the different nuclei, and the different organoids. So this is a multi-scale type of approach where we have both the information about the cells and about the organoids. So how does this data at a certain point look like that you have these individual cells, they grow, and then you have different phenotypic outcome from the same organoid. And so, however, that this is how the experiment look like, and we order it by progression. So this is quite clear for everybody who comes from a single cell genomics background, sense of ordering by progression. But what we do is that we don't use genomic profiles, or we don't use sequencing profiles, but we use imaging profiles. So for every single, and the other difference is that we don't use individual cells profiles, but organoid profiles. So we extract a lot of features from the organoids, imaging features. Then from this, you have individual organoids. So in this graph-based trajectory, every individual point is an organoid with inside the individual cells. And then at the last stage, we can then build this trajectory of growth. And we can be very precise at the decision point. And for example, we were able to determine that the symmetry breaking, so the first Paneth cells, happened exactly between the 62 and the 32 cell stage of organoid formation. So we can be very precise in determining when the symmetry breaking happens. The other part, as I mentioned, is to look at the dynamics, because clearly there we look at 10 and 100,000 of organoids. We can have the full distribution of how the development goes, but really the different interaction between the cells. And so here we built a new light sheet microscope where we can follow the development of this organoid from an individual cells for 5 to 6 to almost 2 weeks. As you can see here, in red are the nuclei, in green is the plasma membrane. On the left, you have the full organoid, and on the right, you have high proliferation. And after a very long expansion of the problem again is how do Gustavo is like multi-lineage dynamics, so to platform where he can follow the individual cells, individual organoid over time, and to create lineage trees. So here you can see this is the lineage tree. We have the first division. That is something that imaging keeps, so we can look at the images and go back and forward. Then we can color code all different type of features, not only of the nuclei, of the cell, but the organoid or the relationship with the different one. So all these approaches, so using highly multiplex, high content imaging with the dynamics, allowed us to have to now really understand how intestinal organoids grow. So what we discovered in the past years is that many different cell types are able to form organoids, so stem cells, but also other cells like Paneth cells or early transit amplifying cells are able to form an organoid. As soon as they start, they have the same exact developmental route. They're going through a stage, this one, this intermediate stage, where there's not a single LGR5 positive cell. These are all YAP positive cells. So there's a full reprogramming of the cells into a YAP positive state. Then to have what we call the symmetry breaking, so the formation of these first Paneth cells, you need to have an heterogeneous deactivation of YAP. And this is very interesting because if we maintain constant YAP, there's no symmetry breaking. If we inhibit YAP everywhere, there's no symmetry breaking. So what we really need in the system is heterogeneity in the individual organoid to then create the notch delta event. Then we were able to show that some mechanosmotic forces are very important for crypt formation, so that the cell fate also drive changes in mechanosmotic forces. And I won't be able to go through the paper very much for today, but the paper is on bioRxiv, and please have a look, because we can combine both single cell measurement but with mechanics measurement. But what we also found, and we found extremely exciting, is that the first step of this organoid formation, so these first 3 days, recapitulate a regenerative process, where YAP serves as a sensor of tissue integrity. So in a way, when you disrupt the cells are individual cells, then you activate YAP everywhere, because YAP is a mechanosensor. It senses, and then as soon around they're alone, they upregulating signaling pathway that upregulate YAP. As soon the tissue is back to be dense, and so the tissue, the cells start sensing the microenvironment and they start to be squeezed, the smaller cells start losing YAP. And the bigger one start maintaining it. So you can create this heterogeneity in YAP by just at the population level. And this is feeding back at the individual cells, enabling the first notch delta event and the first Paneth cell formation. And this is the first step that brings back the system to homeostasis. So YAP sense the need and the transient activation and deactivation. So then we used this, and we found that this was really a very important step. And so Ilya in the lab that started the first day in the lab, so this is a project that started 5 years ago, and this is the paper Fabian was mentioning. So Ilya had the cover of Nature 2 days ago with these gut profiles. And so what he did...
Oh, I missed the cover part. Sorry, I just looked online. Cool. Sorry.
It's cool, and the image is beautiful. So this is, we were very happy about this image. So will be also probably printed for the office, but then it will be later on. So what Ilya did in here was that he took a compound library of around 3,000 compounds, and this is in an arrayed fashion. So every well has organoids with individual compounds. Then he has an iterative imaging where he take a low resolution scan, he segment them on the fly, take the position and goes back and then re-image the organoid at high resolution. These were around 450,000 organoid and 120 terabytes of imaging data. And from all of these, he has been segmenting the organoid. What we did in this case, we didn't do a multiplex assay. We just looked at the two main cell types that are the Paneth cells in the crypt and the enterocytes in the villus that are very spatially resolved. And from these, we have the segmentation and we extract a lot of different features for the same organoid. So here, the first question we had is already how many phenotypes exist? We see a lot of variation, even in the control cell, you don't have exactly identical organoid. And so first question was, how many phenotypes do we have? And here again, here you see a t-SNE of individual organoid color-coded by the different features. And then using a phenocluster from Dana, but also the methods, so we were able to go from a variability in phenotypic responses, so this is the full variability, to what we call the phenotypic landscape of organoid development. And in this case, we have 15 subclusters that are different gradients on strength of different main phenotypes, as you can see here, that are more mature organoid, that are the normal one that we see. We have enterocyte reduced, and then all the different ones until this enterocyst, that these are absence of symmetry breaking. This one is one of my favorite, and then you will see why. This is what we call the regenerative phenotype. This is the phenotype that really looks like an overactivation of YAP. So from this, the other part that was very important, and this is where the key point came out, is like, how do we take this variability and these phenotypic signatures and all this cluster to the compound level? So what we did is that for every compound, created a vector of the distribution of the different phenotype and the phenotypic classes. So now, for every compound, we have a fingerprint or their distribution along the different phenotypic space. And this allowed us to infer genetic interaction and functional interaction from an individual screen. Because normally for these screens, you need to do the double knockout or a parallel screen. But here, using this variability enabled us to create this network. And here we use a method that I developed during my postdoc, but I won't go in detail in it, but what we were able to create is a full network of mapping the genetic interaction. You see this is very modular, especially in some different phenotype. We have high hierarchical upstream regulator where there's beta-catenin and some already known, but also some new component. And what we start looking at, because clearly that was one of our main interests, was this regenerative state. So here we have PPAR signaling, some misregulation in cancer, as known when there's over regeneration, you have some problem in cancer. But two of the main, and this is how the phenotype looked like, but two of the main components in here were RXR and RAR. So these are two compounds that are important and are inhibiting two heterodimers in the nucleus that are responsive to retinoic acid. And they both are in there with very strong penetrance. So we decided to look a little bit more into the retinoic acid and especially the RXR inhibition. And what we saw here, and I will show just part of the data to not show everything, but that clearly in here, so here you see retinoic acid, YAP is completely gone from the nucleus. In DMSO, in control, at a certain point you have this positive YAP cells, but in RXR, in these undifferentiated regenerative cells, you have a very strong increase of nuclear YAP. And what we have in here is that we see that it's also depending on the metabolism of vitamin A, because retinoic acid is not able to be synthesized by cells. So retinoic acid is metabolized by cells by LDH1A1, that is an enzyme here, from the retinol and the diet, so vitamin A. So we see that there's a balance between retinoic acid and vitamin A in balancing a YAP regenerative state to a differentiation state with, in this case, you see it with the retinoic acid. So I won't go into the full mechanism of this for sake of time to show some more unpublished results at the end of the talk, and this is online. But I think this is a very important step, and because we managed to see that YAP is not activated by retinoic acid inhibition, but it's maintained in the nucleus. So we are affecting with these dynamical processes only the translocation of YAP from the nucleus to the cytosol. And this is important because if you give a YAP activator to a mouse, then you start there are tons of highly proliferative tissue that will start proliferate more, that will start activating YAP. However, if you give this RXR inhibitor, nothing is happening, only in the regenerative cells. So this is why we designed in vivo, in a mouse, such an experiment. So we irradiated the mice and we damaged the intestine. And then we treated the mice for a few days with this RXR inhibitor. And the mice recovered much faster, their body weight changes. And if you see here, this is an intestine in vivo. Here are the destroyed villi that are very small. However, in the RXR inhibitor, you have huge expansion, and then there's a very strong increase in cellularization. So the tissue is able to regenerate much faster without having any issues in the control in the unperturbed condition. So to give you a bit of a summary for this part, what we were able to do is start from arrayed multi-well plate based imaging approach, where we could have around 3,000 compounds. The main step were to define the phenotypic landscape, that I think is still one of the most interesting aspects, even on the self-organization part. So how many phenotypes this system is able to create. Then we define the compound profile, we select the target, and we build a network. From there, we were able to go first on the mechanistic level, combining different imaging and perturbation study with the transcriptome, and we see that this effect of retinoic acid on the regeneration is regulating the translocation of YAP and not its activation. And this allowed us to go all the way to in vivo, where we have no issue on unperturbed mice, but we are able to improve intestinal regeneration in very short period of time. And again, the RXR sustain a regenerative phenotype without promoting it. And I think with this, I showed you that we are using the intestinal organoid in a very interesting way, so we can get at the core of some molecular mechanism driving this process. We see that the system is very plastic, so we can not only study how stem cells, but also how undifferentiated cells dedifferentiate to create an organoid. We can mimic and can in a way bottle a regenerative state in 3 days. And this is quite incredible to see that we can look at process of initiation and the stopping of the regenerative process. We can look at symmetry breaking and how heterogeneous population of cells, in a way 16 cousin cells and only one differentiate into Paneth cells. And we can look at also how the different feedback between mechanics and differentiation are involved. The last step is the retinoic acid and RXR, and I think this is one of the first screen and compound-based screen on organoid where we can directly translate it in vivo and look at the regeneration. And I think this would be very interesting to look at in more detail also in human organoids. And I think that when I also was able to show you how to go at the core of decision making across biological scales when we combine some time-course multiplex image with long-term light sheet and high-throughput screen with a lot of also computer vision and image processing, we are able to get at the core of the decision making and understand how these cells take decision. But clearly, the system I showed you is quite simple. We are speaking about one individual cell that goes into an organoid that has maybe 300-400 cells at the end, and maybe a handful of cell types. However, development and also regeneration has a more complex system, and currently there are more and more complex in vitro developmental and regenerative systems that are used to understand how this process are regulated. And so we were a bit challenged by these systems, and so we decided to start with one of the systems that are currently being developed and used a lot, and these are the gastroloids. So gastroloids are these 3D multicellular systems that are grown from 300 ES cells. They can be grown from mouse ES cells, also from human. They have been recently published also on human. We work currently on the mouse. That is the most robust protocol. What happens in here is that in this 5 to 6 days, you have a first step of aggregation, homogeneous population, then you have symmetry breaking and the formation of the 3 major axes. Then after this, and that happens after this Wnt activation. Then what we are able to recapitulate, and what people have been able to recapitulate, is to look at the cellular movement and axial elongation at the germ layer specification. And they are like really correct temporal and sequential gene expression over time during the gastroloid. And recently they also have shown that they can have early heart and gut development. This is, again, we have these 5 days, but you see already the size. This is more than 100 micrometer, almost to the millimeter. These are very big structures that contain a lot of different cell types. So first we need to set up all the imaging around it. So we have, again, as with the organoid, different gastroloid features. We also look at the different intensity on the different axes. And then we divide the gastroloid in 100 segments that can be then measured, and then we can have these long feature vectors. So in this case, the feature vector is really how the different cell types are aligned along these gastroloid. Clearly, this is interesting, but we have initial stage where these gastroloids are very round. So we needed also to set up a different type of analysis where we look at the superpixel and that pseudo single cell segmentation, where we can have the different cells, as you can see here, and we can look how they are organized in this sphere. And then depending if something is all organized homogeneously or has some spatial organization. And we started with the time course, as we do often. Here you see that there are 3 main different, if not 4 main steps. So as I said, is the first step of aggregation, where you start having this homogeneous population, where things are quite homogeneous, where these first initial cells come together. Then what we see is that already at the 48 hours, there's a first asymmetry, where there's some organization and spatial organization of different components of the gastroloid formation. Then what we believe is that the Wnt pulse really helps making this initial asymmetry much more robust. And over time, after the Wnt pulse, we have the elongation. Again, here we have the problem is that we have 3 main steps we're interested in. The first is the asymmetries. How are these heterogeneity and asymmetries emerging? How is the Wnt activation then making the system robust? And what's the feedback in here? And what are the elongation steps? So what we did is that we designed a screen to be able to address the 3 independently. So you can see here, this is the experimental setup. We add the compounds first at earlier time point before the asymmetries all over the Wnt activation, 1 only during the Wnt activation and 1 only during the elongation. Again, these are done in 384 well plate, and we have a full automated culture state. This is very interesting because then we can start layering our phenotype depending on the time course and the different stainings. So this screen was done fixing and looking at brachyury and SOX1. Here you see the first map, first again, the phenotypic landscape of these gastroloid development. Here are the 9 main phenotypes. There are some where there's absence of symmetry breaking, for example, number 6 and number 9. And this is how the different phenotypic classes look like. So the DMSO is quite arranged here. And then we have areas where we have big gastroloids, Brachyri high cells, Icadiri high, and SOX1 high. There are clearly subgroup of phenotypes, for example, this one here, that are extremely enriched at the 32 time point compound. And now we are trying to understand how these different phenotypes can emerge and what are the mechanisms to drive this phenotypic diversity. And this is then helping us in understand how the basic design principle of tissue organization, this is clearly not an embryo. And this is the fun part for us, that it's not an embryo. We don't have extracellular signaling and extraembryonic tissues, and we are just understanding how the individual cells inside the tissue can organize and spatial-temporally coordinate to create a gastroloid. And with this, I would like to come at the end and then how we are trying to understand these individual cells in a tissue, how we are able to cross biological scales. Clearly, we are still down here between cells, neighboring cells, and population of cells. Even the tissue is a bit further away, but understand how a cell can transmit information forward with waves or morphogens and chemical and biochemical gradients, but also how on the other side, the tissue and the different organs are able to feed back this information to the individual cells to take correct decision and make the system much more robust. And so with this, I would like to thank the lab, especially for the first part, Urs and Denise, that they were pioneering the work on the intestinal organoids and the development of intestinal organoids. Ilya, that with the help of Ludwin, really managed to finish this incredible screen that was quite an achievement. And Gustavo for all the light sheet imaging and the new data that are still unpublished, and especially Marietta and Simon that recently started in the lab that are giving really interesting data on more complex systems. Would like also to thank all FMI facility and all of you for your attention.
Thanks so much. Very cool. Thanks also for sharing some unpublished work with us. Maybe we can stop the screen sharing somehow.
Yes.
And I'm
trying.
looking,
okay, let's see.
Looking forward to taking some questions. So people just put questions in the chat or use the question function. There's a first one from Malte Lücken. Malte, I try to unmute you somehow. I think that might be nice if you just raise your hand. Maybe someone can unmute Malte. Otherwise, I just read. So Malte Lücken, he asks, in your organoid trajectories, you relate different organoids in a single-cell ordering based on common developmental process. How synchronized do these organoids replicate development, and is it challenging to build this ordering, or is the time of imaging already sort of predictive for the stage along this trajectory?
Yeah. So the trajectory in this case, because we start from individual cells, we are quite synchronized. So the cells grow, and for example, at day 3, we have quite an homogeneous population. When we color code the timing on the trajectory, especially at the latest time point, we start seeing the spreading of the different, let's say, speed of growth and the different developmental processes. Looking at already gastroloid, there also we managed to have them quite synchronized in their growth, and they're very well controlled. This is a lot of work we are putting up in the culturing condition, and now also with the human organoid is a bit more challenging. So trying to find condition where this time course are very synchronized. Starting from inhomogeneous initial population, then it's much harder. So in our case, they're very synchronized and they look pretty good. Yeah.
Cool, thanks. There's the next question from Heiko Lickert. Heiko, you should be able to talk now. Let me just try again. Heiko? Okay, it should work now. Heiko?
Oh, it should work, yeah. Hi, Prisca. Congratulations for the nature cover and the wonderful story. I mean, you're really rocking. So the gastroloids are interesting, and I think you know the wonderful Yoshiki Sasai paper on self-organizing optical cups, and from RPEs. So that is all obviously in an organ which develops externally. Now, if you go into gastroloids, you said it. Extraembryonic membranes like visceral endoderm, extraembryonic ectoderm are very important as signaling centers to give the embryo patterning information. So how much self-organization can you get in a gastroloid without having you know, these signaling sources from outside?
So from what we see, is quite robust in the sense we have it. It's maybe spatially a bit less robust. It might be, so clearly we have the wind. So the wind pulse is very important to activate this step and to make it more robust. So I think that a lot of it is self-organized, at least on the initial stage. Then, you know, you see the variation, and also when you look at them, these are not embryos. And so from what I have the feeling looking at the gastroloid is that all the extraembryonic is very important to keep it robust, more than defining what cells would do. And having like a positive feedback in reinforcing what the cells would do in any case. So you might have areas where the cells are denser, and then they would create their own microenvironment to then keep a certain cellular fate or differentiate or starting migrating. And these are properties that the cells would do in any case. However, all the extraembryonic part would make, and the shape, depending on the different organisms, would make it much more robust and have the feedback from the tissue to the individual cells.
Great, thanks. So one quick, so do you get actually a Spearman organizer? Do you get a node and a notochord?
If we get, repeat, I didn't hear.
Spearman organizer, do you get a node and a notochord, and is it in the midline? I mean, AP axis, I can understand that you get it by self-organization.
We discussed that yesterday. There might be something in there. We need to look at more gastroloids. So the AP is very clear, and we know exactly when it starts and everything. The other ones we are looking better and seeing, because now we have a protocol that is very robust. We have 100% of gastroloid formation. The timing is very good, so we can go back and also looking at the other one. But I agree, we have been discussing, this is a great question. We have been discussing it really last week and trying to add some more.
Great stuff, Prisca, congratulations.
Thanks.
Thanks, Heiko. There's a bunch of more questions. Maybe I just read one more from the chat from Niklas Bäumer. He also agrees that it was an exciting talk, and he asked how you define phenotypic features of the organoids that you used for the t-SNE. Because, you know, here we're used to the omics part, right there. We just do some dimension reduction. How did you do that?
So we first do, we select features, because the problem with imaging features that are extremely redundant. You have shape features, and you have 80 of them that just give exactly the same and might be more or less informative. So Ilya did a lot of work on different feature reduction. So we started also from some more supervised methods. So we knew some of the phenotypes, and so we looked at also these type of matrices and did this different iterative processes to look at robustness of feature, which one would, for example, come up in different methods. So we went through a lot of supervised. Then we also knew some biologically important. We know that the total intensity of a certain cell type has to be in there, or the mean intensity. So these are very important. This is where, for example, also for the gastroloid, we're there on trying to add to see which are the feature really important. These 100 patterns, the different spatial organization. This is something we continue.
So morphological features essentially, right, of the whole organoids, and then also across the different channels if you have fluorescent activation.
Yeah, and of the cell type.
So much. Now let's take an audio question from Jesse Venvliet. I just unmute her. Jesse, could you ask? Maybe Jesse? So I try to unmute you. You raise your hand.
Would be great to get Jesse's question on gastroloids. Sure.
Self-muted.
Oh, we can hear you, Jesse.
No. Huh?
She said something, right?
Exactly.
Jesse, while you try this out, I read one more question. Again, congratulations for amazing talk from Laurent Vouillard. The gut organoid screen, which properties of the compounds were actually having an effect? Are some pathways enriched among the targets of the active compounds? Because in many cases you do know those targets, right? And are they localized any specific way on the interactome?
No, so clearly, so we started with 3,000 compounds. These are mainly compounds that we know the mode of action. So we already had a selected library of, and then we had around 900 targets, because for us it was important to get to the mechanism. So we took all these. So clearly we already enriched for some signaling, for some really known signaling pathways and different druggable aspects. And then we found, so this is all in the paper, and then we enrich depending on the different phenotypes. So we have the PPAR signaling on the regenerative, we have some notch signaling in the highly proliferation of the planet cells, a lot of metabolism, and different aspects that just are coming.
Jesse now also typed in the question, so I just read that. Sorry, Jesse, for the microphone issue. Great talk, as always. You guys seem to know each other. For the gastroloids, despite having a similar morphology, the endpoint, do you have any ideas how much heterogeneity is there in terms of cellular dynamics? So, for example, convergent morphogenesis?
No, so that's it. Like, so the dynamics, we are trying to look at some of the dynamics right now to see how, especially for the elongation, but these is still in a stage where gastroloids are bigger. And so we have a harder time to get inside, differently than the intestinal organoid that are monolayer. These are filled cells. So we are trying to have different ways and to change a bit our light sheet in a way to be able to look at that. There is some hits are for sure very strongly on migration. So there's something on movement that comes, but mainly on the elongation step, not on the initial stage too much. But maybe also something on the elongation, especially how to create this spatial organization at the beginning.
Yeah, I think particularly these movement dynamics or so with, take time series, even though you just have populations or not matched ones, I think could be very exciting too.
Yeah. So we are looking at those over time, both on the time course and especially in some of the perturbation that we're having. Yeah.
There's another question from Karin Rovatin. She's asking if it's possible to deliver drugs just to parts of the organoid.
Yes, we never did it, but that would be great. So would be to have it like in beads that could release it locally, or can be like optogenetics. You have some trapping ways, and then they can be trapped, and then with light you can open them up. But this is more complicated and not for the full part. We can make part of the organoid, so that is also interesting. We can do only enterocytes or only different type of organoids. This could be one way. The other one is more manual to then inject it on one side. So these are things that we tried for the mechanics project, where this is more localized mechanics. The other one is
the big one, right? Like for the 100,000 one, you cannot do this for the huge one, right? So you have to maybe with the beads, right?
Yeah, yes, with the beads could be, and then the problem that then they're random. We cannot have the organoid in on a perfect orientation. Mm-hmm. So the microfluidics would be another one, but again, we need an orientation that is in a certain way. So it would be interesting maybe to have it in a microfluidics type of chamber to see if, like, with a certain type of gradient or a certain type of, we would have a very stereotypic growth of all of them. And I would imagine that there are some aspect of mechanics. So with some microfluidics, we would get them more all aligned in a certain way.
And you could still stick it into your imaging system, even though it's like on that.
Yeah, thinks that nothing is impossible, but Elias tries on the side to try some microfluidics and things like this to see exactly what is the space we can explore there, combining both. But maybe then, for example, on the gastroloid, we didn't do a 3,000 compounds because the gastroloid growth is much more complicated. So there we have something around 8,500 compounds, because it was impossible in a different way.
There's a whole bunch of more questions. Let's not take stick to this for too long, but maybe take 2 or 3 more. This question from, if that's okay for you.
Yeah.
Question from Simone Aprilmon. Thanks for great insights, even fascinating. What were the main key features for distinguishing subtypes or phenotypes detection among each other. So can you comment on the features in the phenograph from Ilya?
Yes, so these were mainly shape, clearly size of the organoids, and amount of enterocytes and parent cells. These were the three main ones. The spatial distribution, we were hoping to have much more in the spatial distribution there, but there was not much. So these are very spatially organized. If they're there, one is on one side, one is on the other.
Nice. Sophie Seidel asks if, regarding the part of the population dynamics, if you see differing population dynamics, for example, cell division rates or something like that, different in the different cell types?
Yes, but we didn't, yes, there's absolutely. So the first image, breaking the cells, subdividing. We also have a movie of Fucci, is quite clear. You see the division, the stop, and then that's the 16 to 32. The thing that we see is that the YAP positive state, so the early stage, are very fast dividing. We're speaking a full cell cycle in less than 8 hours, G1, SG2, so it's quite incredibly fast, the first day of, so the YAP state is very fast dividing.
We did these movies with the sort of branching lineages with Tim Schröder in early days on the single cell level, right? And now you of course now add all this variation on top of that. That's very exciting, I guess. One more audio question, Malte Luden. Malte.
Hi, thanks a lot for your interesting talk. So you mentioned that you can you put a lot of work into find a way to synchronously develop, let's say, gastroloids and gut organoids. Does that mean you've understood all external stimuli that are required for these to develop in a robust kind of way? And if yes, how far are you from kind of going further and developing a whole organ in vitro, if we do know kind of what stimuli are required to develop in this way?
So on the mouse, if I go fast on the mouse, clearly on the mouse, the medium was very well characterized also already before. We had just tweaked a few things. Now, for example, for the human intestine is harder, so we're working a lot in changing that one to take what we learn from the mouse to make a better human organoid protocol. But on the gastroloid, what we have is the basic control, the basic protocol that was done. We just do it in a bit of an automated fashion. The problem with the gastroloids, for example, is that the Wnt stimulation is homogeneous all over. So then you're inhibiting some other pathways, and then you cannot get the full neurodevelopment, because with Wnt you just don't manage to get it. So I think we won't be able only to change the medium. This will be important, especially for later step. So you will see already, for example, for the paper from Matthias Lutolf and Giuliana Rossi, that where they look at cardio development, there they add different factors to really get to the one they want. And this is for the cardio. So you probably will add factors to get the cardio, and then you will lose something else. So from a full embryo, we are very far, because we will be able to then enrich in some subpopulation. While if we start by engineering some different set, that would be nice. So for example, there are systems with up to Wnt, so then you activate a subpopulation of cells with Wnt and the other one don't get it. That might be also very nice ways to create this complex system and enabling like different perturbations. So that is something we're also very interested in the lab. How do you create controlled heterogeneous population of cells? Because it's clearly that heterogeneity is important, but how do you make this control that at a certain point when there's 16 cells, 5 are YAP high, 5 are YAP low, and 2 are YAP negative. And this in a very controlled way over time. So these are words that we are trying, especially in the lab with optogenetics, to have controlled heterogeneous population. And this might enable us to go a bit further, maybe.
That sounds
I was wondering, thanks. I was wondering, how could you model these things also across scales? I think the scale bridging was a big part of you, and I think in your essays you can really beautifully monitor some of these things. And for example, if you want to control now some of those stochasticity, right? I think some of the models that you derive are really G-network type of things because you do those cool perturbations. But do you think it would be useful or maybe overambitious yet to really go across the scale, maybe really have like a, in early days we had those cellular automata ways of people stick in networks and so on, weren't really predictive. Do you think at some point this should be looked at again, or what's your perspective for that?
So even when I started the lab, one of the goals of my major was like making an agent-based model of like this, like little ones, and then trying to full remodel it. And clearly at that time it was also too early. We didn't know what were the basic rules. For the gastroloid, we are still too far, but with the intestinal organoid, especially at the early stage, we are getting enough of it. So we are thinking about it, how to do it, and then and also to add an aspect of mechanics, because there, being YAPA mechanosensor, we need some aspect of mechanics, I think, to get the full model of the 3D. So that is not only gene expression, because clearly, if you look at YAP abundance over time, at the RNA level is completely flat. That doesn't change. What we see is change is translocation of YAP and the YAP target genes that change a lot. So we need to have a model where we can integrate all this information. And but yeah, that's.
I think it would be really nice if you can perturb this thing and then sort of really be predictive. It would be beautiful. I think we should cut here. Prisca, this was a wonderful presentation. I really thank everyone also for joining and for this great discussion. I think at some point we had more than 230 people or so in there. So I think a lot of attention for such a virtual thing. Not so nice that we don't see each other, but I think it's a cool and fun format. Looking forward to continuing this series. Thanks again for coming.
Bye.
Bye.